written over 100 peer-reviewed papers, and her research is really centered on Willapa Bay. So when I say that Willapa Bay is one of the most studied places around, Jennifer is a big part of that. Another person who's a big part of that is Brent Dumbaugh, who's also on this call. And another person who is not on this call is Curtis Regner. But it is really great to have people like that doing this work. And it's crucial to our going forward. We've got a lot of problems to solve. And I think that getting that level of information out is really important. The other thing I would like to say is I really like the fact that Jennifer is putting her students out here to do this. That's going to be our next generation. Go ahead, Jennifer.
All right. We are going to spend the end of today deep in the mud. And I'm going to just begin by explaining why mud is so fascinating to study from a biology perspective. And that is mud, as anyone knows who's walked through it, is sticky and amorphous and shifty, but it is also really structured by the organisms that live in it. So instead of the organisms simply responding to their environment, they actually shape it. And three of the key players in this regard are the centerpiece of the talks that are coming up: eelgrass, which is a flowering plant, and it anchors the sediment because it has underground stems. Oysters, bivalve, they also tend to anchor sediment because they actually change water flow, and they can even help deposit sediments. And then burrowing shrimp, which are a bioturbator. And this means that they actually burrow through the sediment as they are feeding on the organic material and bacteria that live in the sediment. I'm speaking here specifically of ghost shrimp. So a bioturbator is something that by its biological action actually turns over sediment. And you can see that these activities could potentially be antagonistic to the kinds of anchoring activities that oysters and eelgrass provide. I'm not making any value judgments here. I'm just telling you that these are a set of really interesting and important and highly impactful species within the estuary. And you're going to hear some recent research about them. Bryan Briones Ortiz will start off by focusing in on our native eelgrass. Bryan is a graduate student in the School of Aquatic and Fishery Sciences, and he is our genetics expert amongst those of us working in my lab. And he will tell you some new hot off the press research about the population structure of eelgrass and how related it is to eelgrass in Puget Sound, in Grays Harbor, and also within the bay. So he's really species focused from a genetics perspective. Fiona is a rising second-year graduate student in biology at UW. She actually couldn't make it today, so I will present briefly for her. She has been looking at the role of eelgrass and oysters in terms of providing habitat for fish and crabs and shrimp in the bay. So she's sitting there right at the intersection of eelgrass and oysters because she's studying both of them as habitat. Wes sits there at the intersection of eelgrass and burrowing shrimp, and he will tell you about an enormous experiment that he carried out last summer, basically challenging these species against each other to find out who would win in that race against stabilizing and turbating the sediment. And then Elena will finish up with her work looking at burrowing shrimp distributions on oyster beds. And I should say here, Bryan is funded by Washington Sea Grant. Wes has been funded by state and by Washington Sea Grant. Elena got Washington State Department of Agriculture funding, and Fiona has NOAA federal funding to do her research. So we really appreciate the investment that has been made, both in learning more about the Bay and in supporting the careers of these young scientists. Okay, so I get to do just one thing here, which is to provide you, I hope, with a set of new science glasses. I'm really excited to think about the way that scientists have to look at complex data and to hope that you can also share some of that excitement. So when we go out onto the tide flat, we are often measuring many, many different things. There's a lot of stuff out there, and we're trying to track a lot of it. But just as you can put on 3D glasses to see on a screen what is actually in 2D, suddenly in 3D, science also has some ways of changing the dimensions in which we can actually see things, but sort of in the opposite direction. That is, we collect data in huge numbers of variables or huge numbers of dimensions, but we need a way to be able to display those and communicate those in dimensions that we can actually comprehend. And we can't think in infinite dimensions. So science has a way to take all of these measurements and condense them down to visualizations. And 3 of the 4 presentations, the short presentations that you'll see following this, include this strategy for visualizing complex data. What it comes down to, though, is 2 points that are close together are more similar to each other than points that are far apart from each other. And these sorts of visualizations you will see frequently. Hopefully, we will always remind you what exactly a point represents. So for Bryan, for instance, looking at genetics, he has 1,000 dimensions because he has 1,000 different places on the chromosome where he's looking at variability in the nucleotide DNA sequence. So he can't visualize 1,000 dimensions, but he can take that and he can ask, are 2 plants similar in their genetics, or are they different in their genetics? So 1,000 dimensions, and you now have the science glasses to be able to visualize that in 2 dimensions. For Fiona's work, she's been looking at communities of mobile organisms, and that can be up to 30 species. Instead of having to visualize things in 30 dimensions, she's able to use these techniques to be able to actually see things in 2 dimensions, where each point shows samples that have a similar community if they're close together. And then finally, Elena will show us mud itself in this strategy, which we will sometimes refer to as a multivariate strategy. So just remember, 2 points close together mean 2 samples are similar, and points farther apart in these visualizations mean those points are more different from each other. And you now have been gifted with the ability to use your 1000D glasses like scientists. So without further ado, and I'm going to have each of us just turn over this way, I am going to turn over to Bryan. You are up.
All right, thank you, Jen. Hi, everyone. I'm going to share my screen. There it is.
Looks good.
Okay, thank you. All right, so hi, everyone. I'm Bryan. I'm one of Jen's grad students. I am currently in the School of Aquatic and Fishery Sciences. And the topic for my talk today is the genetic survey of eelgrass in Willapa Bay. So let's get started. A key piece of information, hold on just a sec, it's not moving. All right, a key piece of information that biologists and managers are increasingly interested in is population structure of the species. What this means is that whenever you have a species, it is divided across the landscape in these many discrete populations, which right now they all pretty much look the same with this one type of individual. By investigating population structure, we're able to describe the organization of genetic variation within and between populations, which in other words, it just means how similar and how different are populations from a genetic perspective. In this cartoon, genetic variation has been depicted as different colors. So you can see that some of these populations are more uniform and some of them are mixed. Why is this important? Why do we care about this? Population structure can reflect biologically meaningful events. If we couple, for instance, oceanographic information that inform us about currents, we might be able to explain why we're seeing these mixed populations down south as opposed to up north. And it also has implications for management and conservation. We can use this information to identify, for instance, unique genotypes or unique populations that whose genetic materials are not found elsewhere across the landscape, like these like blue population that is found nowhere else in this cartoon. So this information is very important, but unfortunately, we lack this data for some of our we're lacking this information for a local native species, and one of them is eelgrass. Jen already gave you a brief summary of what eelgrass is, but we might be familiarized with this plant. Essentially, we know that it provides different ecosystem services, including nursery habitat, shoreline protection, carbon sequestration, and more. It also supports a wide array of biodiversity, including some commercially important species. But something else curious about eelgrass is that it can display dramatic trait variation. And one of this, a particular trait of interest is the reproductive strategy of eelgrass. This plant can be either perennial or annual. What this means is that the perennial kind on the left reproduces predominantly via cloning, and each one of these nodes is a clone or essentially a replica of the other individual. And this mode of dispersal basically only produces an offspring or a new clone basically inches away from the parent. In the other type, the annual plants actually reproduce mainly through flowering, and you can see here some flowers and which develop into these seeds, which are able to disperse for far longer distances through currents, for instance. So with this information, I am going to describe the objectives of my research at the university. So there are 3 objectives, and the first one that I'm going to do is I'm going to assess genetic variation of eelgrass Zoster marina across Washington state. To address this question, I have sampled 16 populations across Washington state. As you can see here on this map, there are 16 populations which are divided into 5 different regions. These regions are North, Central, South Puget Sound. There's also Hood Canal, and finally the coast, which is here at the very bottom. This one contains Willapa Bay and the neighbor Grace Harbor. So I'm investigating genetic diversity at the statewide level. And essentially, this is a PCA of genetic variation for different regions of that I'm investigating. Jen has already mentioned how the PCAs show the data. Essentially, the closer they are, the more similar they appear. Right off the bat, we can see that the coastal individuals, which are this group on the far left, are very far away in this coordinate space from the other individuals or the other regions, which are the Puget Sound populations and the Hood Canal, which is expected given their nautical and geographical distance. When we zoom in, overall, we also see that there is substantial structuring within Washington. When we zoom in to this particular area that includes the coastal zones, both for Grace Harbor and Willapa Bay, we are able to see that these individuals share a lot of, share a considerable genetic variation in this PCA, we see that these individuals overlap considerably. So they might be more genetically similar. They are more genetically similar. And then my second objective is to investigate the extent of within bay genetic diversity in Willapa Bay. So to address this question, we decided to sample populations that were characterized by either a region, which is either south or north of the bay. It's not actually south. I will show you in the map. And also life history, which is either populations that were characterized by being either annual or flowering and perennial or clonal. These are the populations that we sampled. So it's not really south, but relative south. And in each of these locations, in each of these regions, we sampled 50 individuals. And you can see that these two regions are actually about 11 miles away from each other, whereas the annual and perennial populations in each location are actually at most a couple hundred feet away from each other. When we investigate the genetic diversity of these populations, we surprisingly find that if we look at this PCA, the annual populations are the red and the blue ones, and they are actually more genetically similar than the perennial populations, which are the green and the purple ones. This is surprising considering that the distance between the annual populations is 11 miles, and these are more similar from a genetic perspective than to their neighboring perennial populations. So that's about the within bay genetic variation. And the third objective in my research is to identify potential drivers to population structure. I'm still working on this. All of this graph that I've shown you are still preliminary results. At this point, I am trying to understand which factors are the ones that are driving and producing these patterns of genetic diversity across the seascape. So to conclude, well, I've so far identified that there is considerable structuring between annual and perennial populations. Again, this is remarkable considering the geographic or nautical distance between the north and south populations. It is known that perennial populations, well, I mean, the clones cannot disperse for too far away, and annual populations, even seeds, have been known to mainly fall within 5 meters from the parent. So this is a remarkable find so far. We've also found high connectivity between the neighboring bays, Willapa Bay and Race Harbor, which means that they share considerable genetic variation. And also, as expected, we found low connectivity between Willapa Bay and the Puget Sound populations all over. So yeah, thank you so much. That has been my presentation. I will take any questions at the end of our talks.
Great, Brian. Thank you. Let's turn over now up to me because I am going to stand in for Fiona. Here we go. All right. Fiona's expertise is in Nekton, and this is a term that refers to large mobile organisms. So these beautiful images here show examples of the things that we get to catch when we are out there sampling across different habitats. The big question that Fiona has been asking is how these organisms use the mosaic, really tremendous variability of habitat types that exist in Willapa Bay. The way, among others, that we have been collecting these data are with a beach seine that is pulled out into a circle and then collapsed to chase all those mobile species into the end. The end is then picked up and held in a shallow bucket while we sort through, identify, and measure everything we have caught. And we can catch anywhere from maybe 5 things to actually more than 400 things in a single seine. I'm sure we don't catch everything because some things will slip under the bottom or hide in a little piece of habitat, but it's a really great, fast and reproducible way of finding out what's living in those invisible places that we can't see when the water is up above. So the design of this study has specifically looked at 2 types of structured habitats, and you can see the 9 different treatments in this grid. We have 3 types of habitats, including no oysters, oysters grown on the bottom, and oysters grown off the bottom, either in flip bags or on long lines. And then crossed with that design, in each of those habitats, we can find places that have no eelgrass, have sparse or patchy eelgrass, or have dense eelgrass. And this allows us to look at the separate and combined effects of these habitats on the community of organisms using Willapa Bay when the tide is up. Okay, so here we have another type of multivariate analysis represented. Each one of these points is one place where we took our seine and collected the organisms that were there. And points that are close together, again, represent communities that are similar to each other. Points that are farther apart had really different species present. So the point, well, the shading of points shows you the different habitat types for oysters. And in this case, you can see that in the summer of 2020, these communities were not distinct across these different oyster habitats. All of these colored points overlap substantially. In contrast, in spring, we did see that the organisms using the aquaculture habitats were different in communities than the ones that were using the areas with no aquaculture structure. These same points are also displayed on the right-hand graphs, but they're color-coded to examine the influence of eelgrass. And in this case, both of the spring and the summer analyses showed that the communities were different depending on how much eelgrass was present. And the types of species actually that we tended to find in eelgrass include things like pipefish and shiner perch and sticklebacks, which we know from prior work also tend to be associated with eelgrass structure. So looking at these results, I think one of the things that stands out is that the oyster structure was more important to these communities of mobile species in the spring than in the summer. And there are a couple of reasons for this. One is that the species that are just present in the bay are different in spring than summer. And so they may be a little bit more choosy. The species may be a little bit more choosy in the spring than those that are present in the summer. The other thing that happens in the summer is that the eelgrass itself just becomes overall larger through the season. And so it could be that the structure of eelgrass becomes plays a more dominant role in habitat choice by these organisms. Whereas in spring, the structure of oysters is more important because eelgrass creates a less of a habitat structure. So we don't know about those mechanisms. And we also have another year's worth of data plus sites that our collaborators have been working on in Samish Bay and in Hood Canal and in Tillamook Bay to contribute to this overall analysis, looking at the role of structure in creating habitat for the Nekton that use Willapa Bay and other estuaries. So with that, I am going to turn over to Wes.
All right, cool. I just want to make sure that everybody can hear me first. All right, looks like we're getting thumbs up. The rain has decided to come down where I'm at, so I just wanted to double check that my connection is good. So, all right, I will share my screen. And let's see here. So does everybody just only see my slides? No words or anything? All right, excellent. So my name's Wes. I am a graduate student, or an incoming graduate student, I guess, in the biology department at UW. And thanks for coming to this and listening to my talk. And I'll be talking about an experiment that I did in the summer of 2020, where I assessed the performance of transplanted eelgrass in a really shrimpy area of Willapa Bay. And so, as all of you know, estuaries are really productive environments, and they're at the interface between both rivers and oceans. And this makes these estuaries really dynamic places. Things like depth, temperature, and salinity, among other things, change seasonally here, but they also can change at different points of the day due to the tides. And this makes these areas really dynamic. And that dynamicness helps create different habitats that are suitable for a lot of different organisms, but it also helps support different industries, both commercial and recreational industries. Like I said, estuaries support a lot of different species, but they also support species whose activities strongly affect other organisms in their environment. And a good example of this, which you're all familiar with by this point, are seagrasses, or our native eelgrass, and burrowing shrimp. Both of these organisms are able to manipulate the biological and physical aspects of their environment through the structuring and the activities that they have. So both seagrass and burrowing shrimp can be found in many parts of the world and have been extensively studied. And a lot of studies have also examined the interaction between seagrass and burrowing shrimp using transplant experiments, where we take both species and physically put them in the environment that the other one creates. However, results from these experiments have indicated that there's no real consistent winner in this interaction on a global scale. So for example, in places like South Africa and New Zealand, shrimp have been shown to be the more dominant engineer, while studies done in California and Oregon have shown that seagrass are the more dominant engineer in those systems. However, while other experiments have shown that seagrass is that more dominant competitor in places like California, Oregon, and even British Columbia, we actually lack quantitative information on essentially who wins in this interaction here in Washington, and especially in Willapa Bay, where both of these species are common. So this is really important because, like I said, not only is the native seagrass, Zostera marina, really common in Willapa Bay, but it provides a lot of different services. So it creates habitat for small and developing organisms. It also acts as food for migratory birds, but it can also help prevent erosion and also mitigate wave forces. It also helps with water quality. Burrowing shrimp, on the other hand, manipulate their environment a little differently. So they create these extensive networks of burrows in the sediment. And what this does is it ends up making the ground really soft and spongy. Not only does this provide homes for small organisms as well, but it also brings oxygen down into that sediment and can also affect nutrient cycling in that sediment. As all of us probably know as well, burrowing shrimp are also a huge pest organism for different types of aquaculture, as the softening of these tide flats through their burrowing activities and the resuspension of particles through their active pumping can smother things like clams and oysters and actually cause them to die. And it's also likely that in other parts of the world where these burrowing shrimp are the more dominant of the two competitors, that these same types of mechanisms are what are likely eliminating eelgrass from those systems. And so thus understanding how these species interact in Willapa Bay is important, because this potential loss of eelgrass in the system could actually have really huge impacts on both the ecological and economical services that eelgrass can provide. And so to address this knowledge gap, we did a transplant experiment where we transplanted eelgrass into an area with really dense shrimp within Willapa Bay. And the goal of this experiment was to determine the effects that dense shrimp have on transplanted eelgrass, and also to identify factors that affect the performance of eelgrass in this area. And so this experiment was carried out on the Long Island Oyster Reserve in Willapa Bay, which you can see in the figure on the left in the red circle. And then the particular location on the reserve you can see as the green dot in the middle. We did this experiment between the months of July and September of 2020. And this particular area is really shrimpy, having around roughly 350 shrimp per meter squared. So in July, we transplanted these eelgrass shoots into these 10 blocks within this area, and each block was populated with eelgrass from one of two different source locations. So orange blocks were planted with eelgrass from a nearby sandy habitat, and green blocks were planted with eelgrass from a nearby muddy habitat. And we chose to do this to see if whether or not where we get our eelgrass from would affect the overall performance of the eelgrass in the shrimpy area. So within each one of those blocks, we established 3 plots, which you can see in red. And inside these plots, we set up 4 different patch sizes of eelgrass, a large patch size, a medium, a small, and a tiny. And we did this to determine if the size of the eelgrass patch has any sort of effect on their performance. We then crossed those patch sizes with 3 different eelgrass densities, high, medium, and low, to also determine if the amount of eelgrass in these patches also affect their performance. So overall in this experiment, our largest plots that had our highest densities contained around 500 shoots, while our smallest plots, those tiny ones, with the lowest density contained a minimum of 2 shoots. And all in all, we were able to transplant roughly 10,000 shoots into this area, which was a pretty big undertaking, but it was pretty fun. And for this experiment, our response variable that we used to determine how the eelgrass performed was the population growth of the eelgrass in these patch size and density combinations. And so with that, these are just sort of 3 main questions that we had for this experiment, which was 1, can we even plant eelgrass into these dense shrimp areas successfully? 2, is there a patch size that could potentially improve the performance of eelgrass? And then 3, is there a patch density that improves this eelgrass performance in the shrimpy area? And so now I'll just move on to the results. And so first off, we did find a significant effect of patch size, but before I explain that result, I want to familiarize you with this figure. So on the x-axis, we have our 4 different patch sizes. So large, medium, small, and tiny. And then on the y, we have our population growth of eelgrass in each one of those patches. This dashed line at 1 represents a stable eelgrass population, meaning that the amount of eelgrass in patches don't change from July to September. Values that are below this line would represent a loss in eelgrass in those patches, and values above that line would represent patches that actually gained eelgrass through growth via branching that was mentioned earlier. Now what we're looking at is each one of these black dots represents the average population growth for each patch size, and these error bars surrounding those dots represent 95% confidence intervals. And if these error bars overlap that dashed line, then the growth rate of eelgrass remained virtually the same over the course of the experiment. Whereas if the bars don't overlap that dashed line, then we know that the growth rate is different, and meaning that the population in that patch was not stable over the course of our experiment. And what it turns out is what we found was that for our large, small, and tiny plots, the amount of eelgrass in those plots remained stable over the course of the experiment, while in our medium-sized plot, we actually had an increase in eelgrass in those plots, which is pretty cool. We also found a significant effect of the interaction between our patch density and then our source location for eelgrass, or where we got our eelgrass from. And so this figure is set up the exact same way, except for now on the x-axis, we have a low, medium, and high patch density. And on the y, we still have population growth. Remember from my previous slide that orange represents eelgrass from our sandy source location, and green represents eelgrass from our muddy source location. And what we found was that eelgrass from our sandy source location performed better when planted at a lower patch density, whereas eelgrass from our muddy source location maintained itself over the course of the experiment. The same is true for both source populations at the medium and high patch densities, as we can see that their error bars overlap 1, which means that their populations remain stable over the course of this experiment. And so in conclusion, what does this sort of mean from, what are we getting from this experiment? Well, if we just go ahead and revisit our 3 questions, we can sort of begin to check them off to see what we've learned. So 1, can eelgrass be transplanted into these dense shrimp areas successfully? And what we found was that yes, not only were we able to transplant them successfully, but it's possible to do this at a high shrimp density during the time of year when shrimp activity is expected to be its highest. And one of the reasons why this could have happened is because from other research that's been done involving interactions between burrowing shrimp and eelgrass, that perhaps the presence of the eelgrass and its roots and stems could actually act as some sort of barrier or deterrence to the shrimp in those areas, preventing them from sort of uprooting and kicking these shrimp out. And so 2, was there a patch size that improved eelgrass performance in this shrimpy area? The answer was also yes. Eelgrass planted at a medium patch size performed better than all other patch sizes in our experiment, which suggests that this more intermediate size may somehow incur better transplant excess through accelerated growth. It's possible that this medium patch size was sort of like the Goldilocks scenario, where it provided the right sort of size and amount of resources available for this patch. And it's also possible that the reasons why we didn't see the other patch sizes performing as well is because of the combination of the amount of resource allocation and just the size of the patch sort of made the net gain or net loss of eelgrass in those patches equal, essentially making them remain stable. And then finally, is there a patch density that improved eelgrass performance in dense shrimp? And lo and behold, the answer was yes. At a low patch density, eelgrass from our sandy source location was able to respond more rapidly and grow better than eelgrass from our muddy source location. And it only did better at this lower density versus our other source and density combinations. And this, what this is essentially showing is that not only could this lower density be more beneficial for eelgrass by maybe giving them more room to grow or more resources available to them, but that we may also only see a source effect at this low density, meaning that transplanting eelgrass into an area more similar to where it came from may actually be beneficial when stocking transplants at this lower density. And so thus sort of the overall take-home message from this was that we were able to determine that it is possible for eelgrass to persist in these shrimpy habitats, and that using a more medium patch size, using eelgrass from a source location that is similar to the area you're putting it in, and planting at a lower density are likely all things that allow eelgrass to grow and potentially overcome the negative effects of shrimp in these areas. And that it's because our eelgrass is there and able to grow that they're likely the winners in this particular interaction that is happening. And yeah, that's my talk. I got to thank everybody in my lab because this was a huge undertaking, many days of planting. And yeah, just also got to thank my funding. So that's it.
Great, Wes. Talk about another huge undertaking. Elena, over to you.
All righty, let's talk some more about shrimp then, huh? So I'll share my screen. Can everyone see that?
Looks good.
And is my audio okay? I'm also competing with background downpour noise.
Seems great.
Cool. So hi everyone, I'm Elena Subbotin. I'm a research tech at the University of Washington, and I'm going to be talking about assessing burrowing shrimp density on shellfish beds using a manual coring method. So the overall objective of this study was to assist with developing an integrated pest management plan by answering a key question related to thresholds for control. That being, what is the abundance of burrowing shrimp on an aquaculture bed? And so in thinking about the potential to apply this method for pest management purposes and also for growers to use, there's sort of a subset of questions that follow that up. First one being, Is this manual coring method feasible, and with what tools? Does it matter where on the bed you're collecting samples from? Does the abundance differ between seasons? And lastly, how many samples would you need to take to detect different levels of certainty about abundance? So we sampled at 14 sites during the fall and the spring, and these beds were chosen by speaking with growers in Grays Harbor and Willapa Bay and getting a feel for where they had concerning burrowing shrimp populations. And the aim was just to include a variety of shellfish beds and culture techniques. As you can see from the map, we went to some places with clam culture, oyster on and off bottom culture, and then also some spots that had no culture when we sampled there, but had previously been used for oyster aquaculture. So generally the layout of our samples was to take 5 samples along 4 transects running parallel to shore and the nearest channel. And so we'd usually walk the length and the width of the bed and then just divvy up how far apart to put those samples. We were using a 1/4 meter squared quadrat to mark that sample, and then also a stainless steel coring device that was 12.7 centimeters in diameter and then 36 centimeters in height. So for the coring method itself, it was developed by the Department of Natural Resources, and it involved taking 5 cores at 70 centimeters of depth. To get that 70 centimeters, we were pulling the clam gun out at least 3 times with sediment in it. And so if you look at the figure on the right, we would put the quadrat down, and then we would record information about vegetative cover, shell cover, and oyster lengths. And then we would also do a hole count, take a sediment sample, and then begin the coring, which would be once in the center of that quadrat and then 4 times on the corners there. We were also sieving the top 20 centimeters of sediment from that first core, just so that we could catch any recruits on the mesh. And we also measured the shrimp by size classes. With those sediment samples, we took them back to the lab and dried them down. Then we weighed them before ashing them at 500 degrees Celsius to determine organic content loss on ignition. Then we put them through a rotap machine, which you can see on the far right over there. And that just has a stack of different sieve sizes, and it shakes the sediment through for about 2 minutes, and that separates everything into different particle sizes. All right, so moving on to some results here, we did determine that the time commitment involved in this coring method was manageable. It took 3.5 hours for our team of 4 people to collect the full 20 samples, which equates to 10 to 25 minutes per person per sample. And also we felt that the stainless steel core was beneficial because there were some places where we were encountering a top shell layer, and the stainless steel core helped us get to that desired depth. Alrighty, so this plot here shows mean density of shrimp per 5-core sample on the x-axis, and then mean holes per 1/4 meter squared on the y-axis. And then the site notation there shows an abbreviation for the aquaculture type. And what we see here is that there's a wide range of shrimp densities from virtually none to up to 30 per 5-core sample. However, the majority of sites had less than 10 shrimp per sample, and then anywhere from 0 to 36 burrows in that 1/4 meter squared quadrat. So what this tells us is a given number of shrimp can be associated with a wide range of different hole counts. And it seemed that we were unlikely to find sites with few holes that actually contained many shrimp, but more often than not, we saw many holes at the surface where fewer shrimp were present. And so all in all, using this burrow count metric alone to infer shrimp abundance is unlikely to be accurate, especially where the shrimp densities are lower and where these holes could be formed by other critters like clams and worms and crabs. All right, so we also saw that shrimp distribution did not differ seasonally. Here are 2 dot plots showing the Port Off Bottom culture site that we sampled in November and May. And you can sort of see that the distributions of shrimp density did not differ significantly between those 2 seasons. And so we also learned that there is some heterogeneity in shrimp density across beds. And so that would suggest that it does matter. Your sampling location does matter in terms of getting an accurate estimate of shrimp populations there. So here was another site that had a sort of strong gradient in shrimp densities. The Westport site was bare and abandoned when we sampled there, but it had previously had long lines and on bottom oyster culture. And so the separation in the density gradient here, the lower 10 samples had roughly 2 shrimp per sample, and then the upper 10 samples had more than 25 shrimp per sample. So this just shows that only sampling at that, the lower 2 transects would paint a very different picture about the overall shrimp abundance in that location. All right, so moving on to some of the sediment data. This is one of those principal components analysis that Jennifer oriented you to. And so each of the points here is representing one sediment sample that we took, and then the shape and the color is coding for the site that it was from. And so it's a little bit difficult to see the relationships here. So I want to show you what each of the axes are showing us. And so for principal component 1, it distinguishes where the samples are falling on the x-axis. And if they're further to the left, that means that they had 125 micron sand more prominent, meaning that when it was put through the ROTAP machine, it had a higher proportion of 125 micron sand. And then if it's further on the right, it had a higher proportion of finer particles and also higher organic content after ashing that sample. And so these 3 sites furthest to the right on the x-axis, these results definitely aligned with what we saw out in the field, because these were spots that were more challenging to maneuver through the sediment, because the mud was definitely softer and almost soupier. And so that definitely coincides with finding more finer particles in that sediment makeup. And then with principal component 2, this is distinguishing where the samples are falling on the y-axis. And it's also telling us how much of those larger grain sizes it has. So 250 microns and above. And if it falls lower there, it has a smaller proportion of those. So for example, with South Bay and Bay Center, they're both falling further to the right. They have more finer particles and also more higher organic content, but South Bay had a larger proportion of the larger sand grain sizes, and Bay Center had a smaller proportion. So the key takeaway from this PCA is just that I've circled the sites that had the highest shrimp densities, and it really looks like the sediment type is not consistent across those sites. They're definitely occupying different places in this multivariate space, and if they had been overlapping more, it would tell us that they were more similar. All right, so the last plot I'm going to show you, this is density of shrimp per 5-core sample with proportion of organic material on the y-axis. And again, the sites included in this figure were the largest shrimp densities that I showed you from that last slide. And what this shows us is that there is a significant negative relationship between abundance and proportion of organic material. The trend in the slopes is definitely decreasing. And so this suggests that samples with more shrimp and therefore more deposit feeding activity in the sediment are associated with this reduced organic content. So to wrap up here and revisit some of the questions I mentioned before, we did determine that the manual coring method was feasible and that a stainless steel clam gun was a useful tool. The sieving portion might not be necessary unless you do have specific interest in recruits and biomass. It does matter where on the bed you collect your samples from, because we were seeing a lot of heterogeneity in shrimp density at our sites. And in terms of the abundance differing by season, it appears that any time of year would be fine to core for shrimp detection. And then in terms of samples needed to determine an estimate of shrimp densities, it would be best to space out your samples in areas where an initial surface assessment might suggest that there are shrimp present. And then in using the method, growers could take roughly 3 to 5 samples and get pretty close to an overall estimate of shrimp populations. And if you take up to 10, that would only increase your certainty. Then lastly, in terms of the relationship between shrimp and sediment conditions, we saw that properties varied substantially across shellfish beds, but that was for reasons other than how many shrimp were present, although there was that significant negative relationship between shrimp abundance and organic material, which might suggest that the density of shrimp could have an effect on sediment properties. And then lastly, just looking ahead to other projects similar to this one, it would be important to assess a threshold of shrimp density that would become negative for aquaculture and other organisms present there. And then also to further investigate the relationship between the density of the shrimp causing harm for oysters and the sediment properties that are there. And that is all I've got.
Excellent. I'm just going to take over here and ask a question of everyone who just talked, except I'm not going to ask myself a question. And then also open up to others but try to hold it to about 10 more minutes. Thanks so much for those presentations you all did, in my humble opinion. Fantastic job. So let's start with Bryan. One of the things that is, really fascinating to think about in terms of this annual and perennial eelgrass is how different are the genetics of those two life history types in Willapa Bay relative to the genetic distinctions that you're finding across Washington as a whole. And maybe I'll just insert there that you didn't use any of the annual eelgrass in your whole Washington study, because annual eelgrass actually is quite rare in most places in Washington. Willapa is another special case where annual eelgrass actually appears. But are annual and perennial eelgrass more different, or is the eelgrass of the coast more different from the eelgrass of the Sound?
All right, so thank you for those questions. So the data is very recent. We only got this a couple of weeks ago. So I'm still working through the, understanding the data and the analysis. That is not a comparison I have made so far. I had these data sets are actually different. They address different questions. There is, as Jen mentioned, there is the statewide level interest of population of genetic variation. And there's a separate study in which we conducted reciprocal transplants between these annual and perennial populations to investigate the genetic basis for these, for whether whether they reproduce clonal or via flowering, if there is any genetic component behind it. The results of the experiment showed that there was indeed a significant genetic component behind these phenotypes, these reproductive behaviors. So we just recently got this genetic data that so far the trends to confirm this finding through the experiment that there is this structuring between these populations. So the next steps are to find if there are specific genetic markers that are associated with conditions that might be, allowing these populations to be there with these conditions. But no, so far I have not yet combined both data sets. But as Jen mentioned, I mean, if this population was a perennial one, and the statewide level PCA shows an overlap with Grace Harbor, then I'm assuming the perennial ones are more similar, share more similar genetic variation. But I have not conducted that analysis yet.
I'm gonna actually answer that for us in a slightly different way, though, which is that one thing that we do have are calculations of a number that says how different genetically two populations are. That number is called FST, and it ranges from 0 to 1. 0 means genetically homogeneous, identical, and 1 is the highest it can get for genetic differentiation. Just for comparison, something like a mobile fish like herring, I understand, has genetic differentiation on the order of 0.1 through all of Washington. So that's just sort of a comparison. The Washington study of eelgrass has genetic variation that is between the coast and Puget Sound, an FST of 0.6. So that's a really highly structured population, which sort of makes sense for a flowering plant that's pollinating really close by and dropping its seeds pretty close by. Can't swim. But then the question is annual and perennial more differentiated than that? Bigger FST than 0.6, or is it smaller than that? And Bryan, what's the answer on the FST for annual and perennial? What's the number there?
Yeah, it was very low. Let me just confirm. I can't remember. It was very low for the annual and perennials.
I'm pretty sure it was around 0.07 because I remember being really
yes
in this exact question of whether annual and perennials were more different than the coast versus Puget Sound eelgrass.
Yes. So the difference between annual and perennials is 0.07 versus 0.01 between same life history strategies.
Mm-hmm. And then about 0.6 for coast versus Puget Sound.
Yes, up to 0.58.
Great. Yeah.
Mm-hmm.
So yeah, I mean, what it really says is that there is some genetic differentiation between annuals and perennials, but definitely still within the same species. But the cool thing that geneticists are figuring out is that species can be pretty closely related to each other, but there can be small changes in their genes that make a really big difference for their function. So whether you're annual or perennial is a big deal. Thanks, Bryan. Okay, so Wes, on to you. The big question there. Oh, go ahead.
There's more questions in the chat that I.
Oh, I know, but I'm going to go around to everyone, and we'll pick those.
Oh, okay.
All right, Wes, the big question for you is what happened to the shrimp after you put the eelgrass in?
Right. So in that particular, so spoiler alert, we're still collecting data on this project. So it's not, we didn't just stop at the end of September, but in that particular time block between July and September, I would say that the shrimp population didn't really change a whole lot during that time block. However, when we came back in April and sampled the eelgrass and cored for shrimp again in that area, we found out that almost all of the shrimp was absent from that transplant area. And right now, it's hard to say what is really causing those shrimp to disappear. We can't core inside of those actual eelgrass transplants because it would essentially destroy them. So we can't say that, we can't do that. But one thing that we can say just observationally is that the sediment conditions do appear to have changed considerably between September and April. So when we returned it, so initially the area was pretty sandy and spongy with larger particle sizes. When we returned in April, there was this sort of thick, sort of silty layer on top, and what appeared to be far less evidence of shrimp activity. So yes, the shrimp have moved or died between September and April, but during the experiment itself, the shrimp population remained pretty stable for the most part. So
yeah, this is one of the cases where I feel like the loss of shrimp was a demonic intrusion when it comes to actually carrying out the research. So great. Looking forward to hearing that tract
yep.
more. Okay, and then Elena, for you, why would it be the case that if you have more burrowing shrimp, there's less silty organic stuff around?
Yeah, so based on that deposit feeding activity, they're shaking everything up, moving everything around, changing the sediment conditions, and creating kind of a siltier, softer environment in that upper layer of sediment there.
Great. Yeah.
I feel like I could briefly answer Nicole's question. She asked about the burrowing activity seasonally. My understanding is that in the colder months, the burrowing shrimp just kind of idle out a little bit. And then when it's warmer, they are more active, creating more burrows and all that. And so that's when you would see more burrows at the surface.
Yeah, that's great. I do feel like you all who are maybe not all like in the science realm might be interested to know that scientists do fight about things sometimes. And one of the things that we fight about is whether we can use burrows as an index of burrowing shrimp abundance or not. And actually, there are lots of places where the index works really well and where it has been published to work really well. I think the issue really for the samples that we were getting on shellfish beds is that those are places that at this point have pretty low densities of burrowing shrimp. And they have really different kinds of sediment types as well. So different amounts of wave energy coming in and different ability for burrows to actually stick around or being sort of obliterated. So the time frame of how long a burrow opening lasts is an issue. And then many of these places, when we were coring them, we got these huge worms and clams as well, all of which can also, they need to breathe at the surface, so they make holes there. So I think the variation in sediment type and the overall lower densities of burrowing shrimp in these particular cases just really pulls apart that relationship at the low end of burrowing shrimp densities. But the coring's not too bad, although it's really good if you have a whole bunch of 20-something-year-olds helping out on it too. All right, let's see. Elena, you want to just tackle one more on what the sediment of Willapa Bay actually is? Someone's asking about how much silt is actually in the sediment, how much organic material. And if you might have an intuition there, I also do have an intuition if you want to punt to me.
Yeah, I might end up doing that, but I will say that even in the spots in Willapa Bay where we sampled, there was very different sediment compositions. You know, when we went up to Stackpole, when we went to a clam bed that was further south, there was very different makeup in terms of how much silt was there, whether it was sandier. And so depending on where in the estuary it was, that was definitely dictating what type of organic content and sediment makeup we found.
That's great. And I'm going to just take this on because I also love that we can bring our discussion to a close with a really, again, deep dive into the mud of Willapa Bay. Most of the sandy areas where you might walk out in Willapa Bay will have less than 1% organic content. And most of them will also have less than 1% silt in them. A place that becomes, you might be going in up to almost to your knee, might be around 4 or 5% organic content and as much as 8% silt. And then the areas that are the cake frosting, the suck mud, where you feel like you might actually just be sucked down forever. A lot of those places are pretty far upriver. They have a lot of fine particles and a lot of organic material coming in. And those can be, I think the highest that we have measured is around 9% organic content, and maybe as much as actually up to 30 or 40% silt, really fine particles. So yeah, and we just go down to 63 microns, which means actually our silt and clay fractions are combined. So we would consider pretty much everything below 63 microns to be silt and clay, and then the sands to be in those larger grain sizes. And as Elena said, one of the big surprises was, wow, there's a lot of difference. There's some large sand areas that mostly have the 250 micron sands, and there's some small sand areas that have more of the 125 micron sands. So yeah, I think I'm going to draw our roundtable to a close and pass back to Doug to bring the whole thing to a close. But just thank you again for your attention, and thanks very much to the students for a really fantastic job sharing what they.
Yeah, hi. Really, really nice presentations. Really enjoyed that. Are there any more questions in chat that we want to go after for the short ones? Good.